Hi, my name is Taylor Curley. I work for the Air Force Research Laboratory, and I'm here with my partners, Alex Huff, Sharon Ellis, and Fred Meyer, as we talk to you today very quickly about a project that we're doing where we're using biologically plausible neural networks to simulate guided attention. Our goal for this project was threefold. Obviously, we wanted to use biologically plausible neural networks to run these simulations. But importantly, we also wanted to simulate the cognitive operations that give rise to visual search patterns, which is very difficult to do in connectionist architectures. And we also wanted to replicate some known search patterns that have been shown in the data or in previous studies. The architecture of our model is shown in a simplified form here. First, what we would do is we take a raw image, then we would separate it out into color and luminance channels. Then we would add a couple of constraints, which are not shown here, but I will talk about in a second. And then after that, it goes through hierarchical processing as an image would in normal human vision, until finally there would be an output, which would be either a motor action or a decision. One of the constraints that I mentioned earlier was constraining the simulated field of vision, which we did in both the horizontal and in the vertical fields. We kept it between 120 degrees and 60 degrees in the horizontal and vertical axes, respectively. And we also had a paracentral vision where the pixels or the image itself would be unaltered within the paracentral vision, but it would be somewhat diluted in the outside, which we basically just used white noise to convolve against the representations in the model. A particularly important part of our model was embedding inhibitory dynamics into the Libra architecture, specifically within the units that represent the colors red, green, blue, and also luminance. We used inhibitory gating, basically what you see here, where red cells are inhibiting green, and then blue cells are inhibiting red and green. By embedding these inhibitory dynamics into the Libre units, we were able to replicate responsivity curves that you would see in the human visual cortex, similar to what you see here, particularly for neurons that selectively fire for different wavelengths of the color spectra. We felt that this was a particularly important part of our simulation for reasons that you'll see on the next slide. When you add all those components together, you can then start introducing either static or moving stimuli to the model. In the case of moving stimuli, you can just take a movie and downsample it to the refresh rate that we have that corresponds to how often the Libra model updates. And here, this is just an example of a particularly difficult task in which the model is asked to track two helicopters, but within simulated night vision. In terms of empirical data, we were able to replicate visual search patterns that have been shown in the literature before. Specifically, we were able to look at a paper by Bruce and Sotos in 2009, and we were able to replicate the visual search patterns that they gave in their paper. We were also able to report the gaze priorities, so where the model decided to look first and second, and so on. So this is an early effort, and we're continuing to work on it, but we hope you enjoyed the presentation. Please let us know if you have any questions. Thank you.